Hello, everyone. Welcome back to Hang Education, where we share the knowledge of biology. So, today you will be studying about enzymes with me. So, let's get started. Enzyme. So the chemicals reaction that take place in the cells, they are controlled by a group of proteins called enzymes. So which basically meaning that proteins, enzymes. So enzymes are made up of proteins. So all enzymes are made up of proteins, but not all proteins are enzymes. So which is really important, meaning that every enzymes, they are made up of proteins, but not all proteins are enzymes. And enzymes are also known as biological catalysts. So what is a catalyst? Catalyst is a chemical or it is a substance which speed up the rate of reactions without being used by itself. So this is really important statement. So catalyst is a type of chemicals which can speed up a rate of reaction. So meaning that a rate of reactions can happen faster. So it will speed up a rate of reaction without being used up by itself. So meaning that if it is not used up, what does it mean? So it means we can reuse the enzyme again and again. So it's indicate that enzymes are reusable or unless they get denatured, right? So that it cannot be used up. And another thing that I would like to add on here is that catalyst is a chemical which speed up a rate of chemicals reactions by lowering the activation energy. So meaning that it decreases or it's lower the activation energy needed to start a rate of reaction. So for overall, catalyst is a substance that speed up a reactions without being used by itself. So it can be reused and it's lower the activation energy. So for this, there is a one important concept that we need to cover is a lock and key model. So before I go over this statement, let's get started with a very simple diagram. There will be an enzyme. On the enzyme, there is an active site. So this is known as an active site. So imagine, why do we call this as a lock and key? Because on your lock, there is the place where we can insert a key, right? So that place would be considered active site, and the lock itself is an enzyme. So let's get started one more time. So there is an enzyme and a specific space or a specific place available on the enzyme. It's known as an active site. So that is important. So that active site will come and bind. So substrate. So here is a substrate coming to join. So this is a substrate, so which is also known as key, and this will be a lock. So a key will come to the lock. So a key will come and join to or bind to the active site where they have a complementary shape. So where they have a complementary shape. So a substrate can come and bind to the active site available on the enzyme. And then the reaction take place. After the reactions, after the reactions, products are forms. And as we stated before that, they are not being used up in the reaction. So the enzyme is stay available and more substrate can come in and join. So I'll repeat one more time. This is a very important concept, so which is lock and key theory, or like we can say lock and key model. So enzyme is like a lock, and then it has a specific place where the key can enter. So it's like an enzyme and there is an active site and substrate will come and bind to the enzyme, which they have the complementary shape. And then there will be a reaction take place. After a reaction, products are formed accordingly. Okay, so this is a process of lock and key model. And these are the sentences that they talk about it. So we got substrate. So substrate is a molecules that an enzyme add on. And active site is a space or an area available or present on every enzyme. And the substrate will attach to the active site of the enzyme, and then reaction take place and products are formed. So as I mentioned before, right, when the substrate joins up with the active site, it lowers the energy, so which is the activation energy needed for the reaction to start, so which allows the products to be formed more easily. Okay, so this is about lock and key model. So you must familiar with the terms enzyme, active site, substrate, end product. So this is the important concept. So enzyme not just only speed up the reaction, it can also catalyze reactions when large molecules are built up from small ones and several substrate molecules, they attach to the active site, reaction take place, products form, and then the products leave the active site, and the enzymes can also start another new reactions, okay? But then one thing that you need to remember is for the lock and key model, right? Imagine I have one key, but I cannot go and open all the lock in the world, right? Meaning that the lock and the key, they are specific, which is the complementary shape. So only my key can open a certain type of lock, right? So that is the main concept that you need to be able to like get from this lecture video, because lock and key model is also another important concept in biology. So enzyme, active site, substrate, and the products are released. And another thing that you need to know is that, so this is a process how they talk about lock and key model, right? So after everything, this is a written part, and the previous one is a diagram illustration. So another thing is that the chemicals reaction taking place in the cells, they are known as metabolic reactions, or we can also call as metabolism. So for the metabolic reaction, we can subdivide it into two parts. So metabolic reaction, which we have anabolic reaction and catabolic reaction. So what are they? So they are the type of reaction. So metabolism or metabolic reaction, they are a general terms, and anabolic and catabolic. So catabolic mean breaking down, so meaning that they break down large molecules into small molecules. Anabolic mean building up. So meaning that for the anabolic reaction, they build up from the small molecules to become a large molecule. So example would be when glucose molecules combine together and form into a large carbohydrate. So that would be the anabolic. And as for the catabolic, it's like when we eat rice or starch and then they get broken down into smaller units or smaller parts, which are known as the catabolic reaction. And also another thing that is extracellular enzymes and intracellular enzymes. So as for the extracellular enzymes, so as you can guess from the name itself, so extracellular mean they secrete or they produce the enzyme out of the cell. So it's mean excrete, not excrete, it's more like a secrete out of the cells, and they have the function outside the cells. For the intracellular enzyme, so you can already guess, right? Extracellular outside the cells, then intracellular mean they have a function inside the cell. So this will be about the metabolic, anabolic, and catabolic. So as you can see from the diagram, so for the upper one, so when the two small molecules, A and B, they will come and join into the active site, and they form into a larger molecules or larger particles, so which we can also call as anabolic reaction, right? Because it's building out from a smaller molecules into a larger molecules. For the lower or part B, which is catabolic reaction, since we have a one big molecules and it's get broken down into, it can be two or three or whatever products. So whatever is mean that large molecules into smaller molecules, which mean that it is known as catabolic reaction. Okay, so you have to remember about lock and key model, and there are two types of two component in metabolism or metabolic reaction, which are anabolic and catabolic. So anabolic, it's me building up, and catabolic mean breaking down. So building up from smaller molecules to larger molecules, or catabolic breaking down from larger molecules to smaller molecules. So these are the example that you can like read through for the some of the example that are available in the body as well as for this. So this is the anabolic and this is a catabolic reaction. We can also say catabolism, anabolism is the same thing. And this is the key summary about the enzyme and that we have went so far. Okay, so after we learned about enzymes and lock and key model, and you should also be aware of the factor that affect the rates of reaction. So there are a total of 4 different factors: temperature, pH, substrate concentration, and the presence of inhibitor. But for today's lecture's video, we will be only focusing on temperature and pH. For the substrate and presence of inhibitor, they are specific to human biology, not for the general biology. But whatever, it's also a factor that can affect the rate of reaction. So as for the temperature, so not just reading all the slide, we can go through with the graph. So for the graph, I will be subdivided into three different portions. So numbering 1, 2, and 3. So as you can see from the graph, as the temperature increases, the rate of reaction increases. So this is a part A, right? So you can see that as the temperature increases, the rate of reaction increases. So this is a description. So if we want to explain like why it happen or what happened when the temperature increases, why does the rate of reaction increase, right? So by increasing the temperature, they gain more kinetic energy, right? So for part number 1, so increasing temperature provide more kinetic energy or they gain kinetic energy. So by gaining or by receiving more kinetic energy, they move faster, or if you don't want to say move, you can also say collide faster or faster collision. So faster movement or faster collision mean the rate of reaction will increase because there will be more successful reaction, right? More successful collision. Okay, so that would be the part 1. So where the rate of reaction increases as the temperature increases. And then it will reach to the optimum temperature. So whenever you talk about enzyme, you have to use the words, specific words. In this case, you have to say optimum. So optimum basically means is the best temperature where the rate of reaction is taking place, is the greatest, the best, everything. So it's mean optimum temperature, meaning that the rate of reaction at is the fastest. So that the temperature that the reactions in the body, they are at the fastest. So in for example, in human body, it will be around 37 degree, right? Body temperature. Another interesting part is like what happened when the temperature goes beyond. So, right, I think most of you watching this video may ever have fever or like you may ever have feel sick, right? Where your body temperature go beyond the normal body temperature. So what happened is like when the temperature get beyond its optimum temperature, we said that enzyme is denature. So enzyme is denature. What does it mean by denature or like denaturation? So denaturation is, or we can say denature, or denaturation is like when the shape of active site changes where the substrate can no longer join or no longer bind. So meaning that denaturation means the shape of the active site changes where the substrate can no longer, we can say fit in, no longer fit in, right? So it's mean imagine, right? So from the previous example, this would be the original shape of an enzyme. So this is just an example, right? So normally, so they will come and bind this type of shape, right? So this would be substrate and this is an active site. And then due to the temperature, the shape of the enzyme get changed, something like that. So it's me, the reactions cannot take place because the shape of the active site changes. So the substrate that originally can bind will not be able to bind or fit, right? Because the shape of the active site changes. So that is known as denaturation. So I'll repeat one more time. As the temperature increases, the rate of reaction also increases because they gain more kinetic energy, so they move faster and more successful collision, and they will reach to the optimum temperature where the reaction as is highest, the fastest, the greatest, And beyond the optimum temperature, the enzyme is said to be denatured. Denature means that when the shape of the active site changes and substrate can no longer fit in, and the rate of reaction will drop. So the real life example would be like when we boil an egg, right? So that the color changes. So the white protein, right? The egg white, right? So that would be the real life example, or like when we cook some protein in the real life. So those are the example. And also when you get sick, right? So the body temperature goes beyond the optimum, and then you feel like not doing something and you're not very uncomfortable, right? That's because the enzyme in our body get denatured and the rate of reaction gets lower. So this would be the first factor that affect the rate of enzyme activity. And this is all the detailed explanation written there. And I prefer to explain from the diagram, which I feel it's more obvious and it's more visible, right? So it's visual. Yeah. And so these all are all about explanation. And then another factor would be the pH. So this is also the same thing where we can still use the optimum pH and the denature. But with pH, what with pH that you need to remember is at the either extremes of pH, right? So it can be very acidic, very alkaline, right? So the enzyme can denature. So enzymes, they only work as the specific pH, right? So you can see that for most enzyme in our body, they may work at optimum pH of 7, but depending on the location, right, it may change their optimum pH. But what I want to say is that with the pH, enzymes, they can only perform at their best only at a specific pH. If the pH get lower, so meaning acidic, or if the pH get higher, meaning alkaline, the enzyme is said to be denatured. So these are the few examples that are available in our digestive system. So in the stomach, we have hydrochloric acid. So acid means stomach has around pH 2, and they are enzyme like that can survive and that can do the reaction. For example, pepsin enzyme and trypsin enzyme in the small intestine with around pH of 8, right? So we got acidic condition and alkaline condition as well. So this would be the other factor that affect the rate of enzyme reaction. So we got temperature and we got pH. So that would be the all about summary about the enzyme. So just a wrapping review summary. So you need to be familiar with lock and key model, and you have to know about catabolism and anabolism, so which we combine as a metabolic reaction. And also another thing is a factor that affect the rate of enzyme reaction, so which includes temperature, pH, and the presence of inhibitor and substrate or the enzyme concentration. So that's all for today videos about the enzymes. And thank you so much for watching. And if you have any questions, you can contact me through my email or my Instagram account. And hope you learned something new about biology today, and hope to see you all in next video. Thank you so much and you have a great day. Bye bye all.